Okay, we're going to get started. So this is the Translational Research and Clinical Oncology course, and my name is Terry Moody. And we've been doing this course for about 10 years now. And my office, they just moved me up to Shady Grove. So you see I have an unusual phone number for NIH, but I still do research down here in Building 10. And the best way to get me is just send me an email. So in terms of organizing the committee, I especially want to thank Jonathan Wiest. He's the director of the Office of Training and Education in the CCR, and he pays the bills for this class. And so in terms of the schedule, we always lecture on Mondays, except for the October 13th week. That day is Columbus Day holiday on Monday, so we're lecturing on Wednesday, that particular day. So today I'm lecturing, as well as Jill Smith, about clinical trials. And then we start going into different types of cancer. Next week we talk about lymphoma as well as prostate cancer. The following week, ovarian cancer. We added a new lecture this year on immune checkpoint inhibitors. This is sort of a new area in clinical research that's having great success. And basically, it's been known for a long time that many cancers don't really respond to the immune system, such as my specialty, lung cancer. And it turns out it makes certain ligands that sort of neutralize the immune system. So they are now using antibodies to neutralize these ligands, and consequently, the cancer is more responsive now to immune therapy. And then another thing that we emphasize is in the past decade, lots of small molecule tyrosine kinase inhibitors have been developed, and these are now used clinically in different types of cancer. And then in October, we continue the class, and we focus on tumor imaging. A goal in cancer always is to detect it early when it's in stage 1 and amenable to surgical dissection. And then we go into specialties such as breast cancer. Radiation oncology is used to treat a lot of different cancers. And then in November, we have a lecture on stem cells. Cancer stem cells are very resistant to the traditional types of therapy, such as chemotherapy. And then in the end of November, we go into special topics such as RNA interference, microRNA. We talk about topoisomerase inhibitors. And finally, we end talking about nanotechnology. So,
hi, Terry.
Yes.
Sorry, this is Chris up at Frederick. We're not seeing the presentation right now.
Okay. You'll work on it.
Do you have any? Okay.
Here he comes. So in terms of registration, we have 150 registrants. If any of you want to take the class and you haven't registered yet, see me after class and you can just fill out a form and we'll get you registered. And then one unique component of this class relative to an academic class, we sort of have a hands-on section where you can sign up to visit tumor boards where they review patient cases, and then you can visit various core facilities, such as places where they do next-generation sequencing. So I'll be working on this later this week, and I'll be sending all of you an email to see if you want to sign up for various components, and these usually occur in October as well as November. And finally, you can get a certificate of passing the course. We have a computer-based examination at the end of the course. It's not that difficult. There's sort of one multiple guess answer from each lecture, and to pass, you need to get 70% correct. So this course, all of the lectures are in Bethesda, but we have people at NCI Frederick that we video conference to. 2 days after the lecture, the video cast is then posted on the archive, and you can call this up on YouTube at any time. So if you miss a lecture on Mondays, 2 days later, you can just call it up on your computer and see it there. Are there any questions on the organization? Okay, we'll get into the meat of the lecture now. And that being, we first get an overview of the different types of cancers that are important in the U.S. And of course, lung cancer kills about a third of all the patients that die from cancer in the U.S. Annually, we have an incidence of about 170,000, and almost 160,000 are dying. So this means that our treatment of lung cancer needs to be dramatically improved. And a little later on, we'll be discussing tyrosine kinase inhibitors that are coming into play in the past decade. In contrast, colon cancer, breast cancer, prostate cancer, it's much more amenable to treatment. You see for breast cancer, we have a little over 200,000 being diagnosed each year, but the mortality rate is a little less than 40,000. So we're getting quite good at treating breast cancer now. We've developed lots of antibodies as well as tyrosine kinase inhibitors and various chemotherapeutic drugs. So lung cancer is very difficult to treat. Total, we have about 1.3 million cases of cancer diagnosed in the U.S. annually, and about 550,000 people die annually from the disease. But what's going on is as our treatment gets better, we get more and more cancer survivors. And now we have over 12 million cancer survivors in the U.S. So we mentioned the big four, the lung cancer, colon cancer, breast cancer, prostate cancer, other cancers that kill 10,000 to 30,000 annually include pancreatic cancer, which Joe will talk about shortly. And again, pancreatic cancer is very difficult to treat, as is ovarian cancer, and then brain cancer, the glioblastoma. So we have four cancers then that are very difficult to treat: the lung, the pancreatic, ovarian, and the glioblastoma. So these are areas where we really need to improve our treatment. And several risks have been identified, such as drinking excessive alcohol. This can contribute to liver cancer. Asbestos, which is in the insulation of a lot of walls. This causes mesothelioma of the lung. The diet can cause cancer, such as I just came back from a meeting in Japan. And in Japan, they eat lots of pickled food, and as a result, they get a higher incidence of stomach cancer. That's quite rare, really, in the U.S. There are genetic factors, such as in breast cancer. We have the BRCA1 and BRCA2 genes, which when they get mutated, the women then get the breast cancer. Various hormones come into play, such as estrogen for women, and that can induce breast cancer. And for men, we have the testosterone, which can contribute to prostate cancer. In terms of obesity, a high body mass index is associated with increased risk of colon cancer. In terms of radiation, ionic radiation leads to leukemia. So when we dropped the atomic bombs on Japan in the 1940s, many people died from leukemia. Tobacco, of course, is a risk factor for lung cancer. UV radiation can cause melanoma. So in our southern states, Arizona, Texas, Florida, we have a very high incidence of melanoma. And then viruses can contribute to various cancers, such as the hepatitis virus that causes liver cancer. This is quite high in China. And then the cervical cancer, the HPV, can lead to cervical cancer. So we have many risk factors then that are associated with cancer. So my specialty is lung cancer. We'll sort of briefly give an introduction to that. So it kills only 150,000 patients annually, and it's associated with cigarette smoking. There's 45 million current smokers, 45 million ex-smokers in this country. But of these smokers, only really 16% will die from lung cancer. Many of them will die from other things, such as heart disease. So in terms of the carcinogens that are present in the cigarette smoke, there's many that have been identified, such as heavy metals, chromium, but then we'll focus on the polyaromatic hydrocarbons, the PAHs. Also, the NNK is a factor associated with lung carcinogenesis. So in terms of the polyaromatic hydrocarbons, they're in the cigarette smoke, but then they have to be activated to become a carcinogen. In particular, they have to be oxidized, and then they form adducts with the DNA, causing mutations in the DNA. And we see we have GC going to TA mutations. And two genes frequently mutated in lung cancer are P53 as well as KRAS. So benzapyrene, you see it's an aromatic hydrocarbon, lots of six-membered benzene rings in it. And here's how it's activated by P450 enzymes as well as epoxide hydrolase to the active carcinogen, which is BPDE. Then this binds to the DNA, especially guanine nucleotides. And so the DNA gets mutated, and P450 enzymes will then try to detoxify the carcinogens, and in particular the phase 2 enzymes will try to convert the carcinogens into water-soluble products that can then be excreted. But the DNA gets mutated if the rate of carcinogen activation exceeds the rate of carcinogen detoxification and or DNA repair. So with lung cancer, the problem is people smoke cigarettes, and after about 20 years, then they start getting cancer cells in their body, and more genes get mutated. Really, dozens of genes get altered in lung cancer. So P53, it's mutated in about half of the lung cancer patients. And what we see is P53, it mediates the G1 to S phase checkpoint of the cell cycle. And if the DNA gets mutated, it tries to induce programmed cell death so that the cancer cells will die. But when the P53 gets mutated, then it becomes inactive, and it can't cause the cancer cells to die. So G to T transversions occur at CpG-rich codons. And you see from this slide that certain codons are especially mutated, such as codons 157, 158, 245, 248, 249, and 273. So the mutation is not a random process, it occurs at specific areas. So then in the cell cycle, the P53, it regulates G1 to S phase transitions. G1 is the resting phase of the cell. The S phase is where the DNA is replicated. Then in the G2 phase, additional enzymes are made. And then in the M phase, the chromosomes are segregated, and the parent cell becomes two daughter cells. And then we go back to the G1 phase. So in certain cells, such as epithelial cells, they can become quiescent and rest in the G0 phase. Cancer cells don't do that. They just grow as fast as they possibly can. So we mentioned that P53 mediates the G1 to S phase checkpoint of the cell cycle. When the DNA gets damaged, then P53 tries to be increased, as well as P21, which is an inhibitor of the cyclin-dependent kinases. And normally p53 drives programmed cell death or apoptosis after DNA damage. But if the genes are mutated, this does not occur. So in the cell cycle, then, we have a whole series of enzymes regulating it. At the G1 phase, the cyclin D1 and cyclin E are important. In the S phase, the cyclin A is important, as well as the G2 phase. In the M phase, the cyclin B is important. In the G1 phase, the cyclin-dependent kinases 4 and 6 are important. In the S phase, the cyclin-dependent kinase 2 is important. In the G2 and M phase, the cyclin-dependent kinase 1 is important. And the cyclin D, it's inhibited by many factors such as P21, P27, P57, P15, P16, P18, and P19. So there's a whole bunch of cyclin-dependent kinases, as well as cyclins and cyclin-dependent kinase inhibitors that are involved in the process. So we mentioned that in terms of tobacco smoke, we measure tobacco smoke in terms of pack years. If you smoke 1 pack for 1 year, that's 1 pack year. Most chronic smokers smoke 2 packs each day for a year. And then after 20 years, that would be 40 pack years of cigarette smoke. But we see after 10 years, the lung starts to undergo hyperplasia and metaplasia. After 50 years, dysplasia can result. And then after 20 years, a carcinogen in situ can form. And after 25 years, a malignant cancer can form. So the cancer cells then are constantly mutating. And the cancer becomes increasingly aggressive. So we see here a little cartoon of the normal cells in pink. With hyperplasia, there's more of the cells. With dysplasia, the cells start to disform. A carcinoma in situ, we start seeing some malignant cancer cells forming, but they're still confined to the primary organ, such as the lung at this point. But then when a full-blown cancer develops, it can undergo metastasis to other organs. And the lung cancer undergoes metastasis to the liver, lymph nodes, bone, and brain. So this is just an H&E stain of the lung tissue. And in pink, you see the cytoplasm. In purple, you see the nucleus. And so in this case, the lung is highly ordered. And on the surface of the lung, we see little villi. And that's where the air is exchanged. We breathe in O2, exhale CO2. When we get to hyperplasia, now we see more and more cells. It's not an orderly layer. The layers now have 5 or 6 cells in them. And then when we get to dysplasia, the cells start to become disordered, and we see the villi now are starting to disappear, and the nuclei are becoming very big. Cancer cells have big, dark nuclei, scant pink cytoplasm. And this is then an adenoma of the lung. So in this case, these are sort of this is like a carcinoma in situ. And then with further progression, we can get a full-blown adenoma, and this secretes lots of mucus. So there's these islands of mucus around the bodies of cells, and then these cells can undergo metastasis to distant organs. So as the process of carcinogenesis increases, we see more and more purple nuclei, less and less cytoplasm, pink. And here's another cartoon showing we have the carcinogens. It says the benzapyrene, and some cells get initiated. And then tumor promoters, such as growth factors, induce these altered cells to grow. Then you get a carcinoma in situ, and with further genetic change, you can get a red carcinoma forming that can then undergo metastasis. So Initially, we have a carcinoma, a little cluster of tumor cells. But then as the tumor gets big, what happens is it needs angiogenesis, whereby blood vessels from the host will then grow into the tumor. And these will provide oxygen for the tumor and nutrients for the tumor for further growth. If this does not occur, the tumor cells in the interior will just die. They'll starve. They won't have oxygen. They won't have nutrients. So the host actually sends its blood vessels into the tumor. And then the tumor further grows, and it can undergo migration, invasion, and metastasis. And Dr. Zedere will talk a little bit later on in the course about the process of angiogenesis. And genetic abnormalities, we can get mutation of tumor suppressor genes, such as p53. We don't always need the genes to be mutated, though. We can just silence the genes through epigenetic phenomena. And Dr. Verma will talk a little bit later about this in the course. And then you can get amplification of oncogenes, such as c-MYC, a nuclear transcription factor. Cyclin D1 we saw was a protein involved in the cell cycle. We'll be talking shortly about the EGF receptor, which is a tyrosine kinase receptor, as is the ERB2. So in terms of tyrosine kinase receptors, these are big proteins with an exterior outside the plasma membrane. This is where they bind the various growth factors. And then on the inside, we have a large tyrosine kinase domain. And what this will do is it will find tyrosine substrates in the tyrosine kinase receptor or in other proteins, and it will cause tyrosine phosphorylation of these proteins. Then the proteins will become active and alter second messenger production. So we not only have the EGF receptor, another one that's important is the IGF-1 receptor, insulin-like growth factor 1 receptor. The fibroblast growth factor receptor is important. And the VEGF receptor is important in endothelial cells. So the cancer cells make VEGF, and then it stimulates the endothelial cells to grow into the tumor and cause angiogenesis. And so we'll be focusing here on the EGF receptor, which has many ligands it can bind. Most cancer cells make transforming growth factor alpha, but also heparin-binding EGF is another factor that many cancer cells make. And we see we have several names for it, the EGF receptor, or it could be called ERB1 or HER1. The EGF receptor is very important in lung cancer. In breast cancer, the ERB2 receptor is important. But another name for it is HER2 as well as NU. And then other members of this family include the ERB3, which binds erregulin, and the ERB4, which binds NRG2 and NRG3. So what happens with these tyrosine kinase receptors is once they bind the ligand, they then dimerize. So they can dimerize with itself. EGF receptor can dimerize with itself, or it can form a heterodimer with the HER2/neu, and then you'll get not only the EGF receptor being tyrosine phosphorylated, but HER2/neu will be tyrosine phosphorylated as well. So we mentioned the EGF receptor. It's a big protein, 1,186 amino acids. On a Western blot, its molecular weight is 175 kilodaltons. We have 621 amino acids in the extracellular domain, and the extracellular domain binds EGF or TGF-alpha. Then there's a single transmembrane domain, 24 amino acids, and then there's 541 amino acids in the intracellular domain. And lysine 721 binds ATP, and then it will transfer the phosphate to a tyrosine amino acid. And various proteins can be tyrosine phosphorylated, but then the EGF receptor can be tyrosine phosphorylated as well at 4 different sites. And here we see that the EGF receptor binds with high affinity, as does TGF-alpha. And Ira Paston at NIH, he makes these growth factor Pseudomonas exotoxin 38 conjugates that are cytotoxic for cancer cells. Monoclonal antibodies, though, have been also developed for the EGF receptor, and these are used to treat especially colon cancer. And here we see the EGF receptor getting tyrosine phosphorylated. It's a very dark band. But also, phospholipase C can get tyrosine phosphorylated, as well as phosphatidylinositol 3 kinase. And these participate in second messenger production. So for the EGF receptor, what's really unique about lung cancer is it gets mutated. And two areas that are very commonly mutated are the L858R and the G719S. When these get mutated, then they bind tyrosine kinase inhibitors with high affinity. And the tyrosine kinase inhibitors that are used is gefitinib in Europe and erlotinib in the United States. So these are approved by the FDA in the US for treatment of lung cancer patients who fail chemotherapy. And here we get another look at the cytosolic domain of the EGF receptor, and we see the mutations, the G719C and the L858R. There's also some deletion mutations. And then these make the EGF receptor sensitive to the tyrosine kinase inhibitors. So what we've done now is we're at the age of molecular medicine, and what to do now in NCI protocols is if a lung patient comes in, they'll do a molecular analysis to see if the EGF receptor is mutated. And if it is, then they know they can use these tyrosine kinase inhibitors on the patient. So in terms of second messenger production then, when the EGF receptor gets activated, it can induce proliferation of the cancer cells through this pathway called the RAS, RAF, MEK, and ERK pathway. So KRAS, one of the initial steps, it's mutated in about 20% of the lung cancer patients. And when it gets mutated then, normally RAS will bind GTP, but then it will get hydrolyzed by a GTPase to GDP, which is biologically inactive. But when the RAS gets mutated, the GTP remains bound. And in particular, the mutations are usually G to T at codon 12. And right now, the Frederick National Lab is doing a big project with RAS, trying to come up with new modes of therapy for patients who have mutated RAS. RAF is downstream from RAS. Once RAS gets activated, RAF can get phosphorylated, and then it becomes biologically active. And with RAF, it's actually mutated in melanoma. So we have the mutation, the V600E, and when this mutation occurs, then you can use this tyrosine kinase inhibitor, the PLX4032. And this is very effective on the patients initially. So both RAS and BRAF, when they get mutated, they're what's called driver mutations in the cancer. So we stated that in lung cancer, you have dozens of mutations, but not all of them are what's driving the cancer. If you have RAS getting mutated, however, or BRAF, that's a very serious event. And then you can target that in your therapy. So once RAF gets phosphorylated, it then will phosphorylate MEK. And there's two types of MEK, MEK1 and MEK2. And they have inhibitors for the MEK, such as the selumetinib. And this is being investigated in therapy with the chemotherapeutic agent docetaxel in KRAS mutant lung cancer patients. And then downstream from the MEK is the ERK, and when the ERK gets tyrosine phosphorylated, it goes into the nucleus and alters expression of transcription factors, such as FOS or MEK. So this first pathway then, it's very important in terms of proliferation. And here we see the EGF receptor dimerizing. And then when it dimerizes, it causes GTP to be bound to the KRAS, which will then activate the RAF, which will then phosphorylate the MEK, which phosphorylates the ERK, which then goes into the nucleus and alters the expression of transcription factors. So what we do is we get signal amplification from this. Just activating one EGF receptor, you'll get thousands of phosphorylated ERK. And this process occurs very rapidly. In the lab, I can add EGF here, and within 1 minute, I can see phospho-ERK. So it's a very rapid process. And here is a cartoon sort of showing the EGF receptor. And here we just see a monomer. But then once the EGF binds, it starts to dimerize. Then it will undergo a conformation change and turn on the tyrosine kinase activity. And a second big pathway for the tyrosine kinase receptors is the PI3 kinase AKT-mTOR pathway. And this causes survival of the cancer cells. So the ERK pathway causes proliferation. Here, we minimize the apoptosis of cancer cells. And so what happens is when the EGF receptor dimerizes, then we get PI3 kinase, and it produces the phosphatidylinositol phosphate metabolites, which then in turn stimulate the phosphorylation of AKT, which causes the phosphorylation of mTOR. So the PI3 kinase, the 100 kilodalton subunit, is involved in the metabolism of the phosphatidylinositol, and then the PI3 kinase can be mutated in breast cancer, about 25% of the patients, glioblastoma about 25%, colon cancer about 25%, and stomach cancer about 25%. When it's mutated, then it's turned on, and so you get more phosphatidylinositol metabolites. PTEN is an enzyme which tries to metabolize the phosphatidylinositol metabolites, but it's mutated in about 13% of the breast cancer patients. So when PTEN's mutated, then the phosphatidylinositol metabolites build up. And then AKT is designed to prevent apoptosis of cells, and in particular what it does is it phosphorylates BAD and caspase 9, making them inactive, so this prevents apoptosis of the cancer cells. AKT, it's mutated in about 5% of the breast cancer and 6% of the colon cancer patients. And finally we get to mTOR, and mTOR, one of the key things it does is it causes autophagy of cells in a lysosome-dependent pathway. And if it's not active, then there's decreased autophagy. And this is a clinical protocol that's being done at the NCI. And so this is for lung cancer patients. And initially what's done is they do a biopsy of the tumor to see what genes get mutated. And then if the EGF receptor is mutated, the patient is treated with erlotinib, a tyrosine kinase inhibitor. If the EGF receptor is normal, they then look for RAS, RAF, or MEK mutations. And if one of these get mutated, the patient is treated with the MEK inhibitor, AZD6244. If that's not mutated, they then look for PTEN mutations, PI3 kinase mutations, or AKT mutations. And if they see that, they treat with the PI3 kinase inhibitor, MK2206. They then look at ERB2 mutations or amplifications. If they see that, they'll treat the patient with lapatinib. They then look for KIT mutations, PDGF mutations, PDGF receptor A mutations, or amplifications. And if they see that, they treat with the tyrosine kinase inhibitor sunitinib. And if that doesn't work, they try to figure out something else. But then a key thing is with these tyrosine kinase inhibitors, they find that if they give them to patients, the patients can become resistant to the drug. So at the end of the study then, if they see resistance, they do another biopsy specimen and see if the tyrosine kinase enzyme has been further mutated. So this is the age of molecular medicine, and so we do all of these analysis, and what we see then, based on the mutations, dictates the therapy. So we mentioned that in about 50% of the patients, they develop resistance to erlotinib and gefitinib after a year. And primarily what they see this is due to is a secondary mutation of the EGF receptor T790M. So another drug that's been used in chronic myelogenous leukemia patients is the tyrosine kinase inhibitor gefitinib. This was done in the early 2000s, and it's what started the craze of tyrosine kinase inhibitors. And for the CML patients, they normally have an alteration of chromosome 22 called the Philadelphia chromosome. Its segments of chromosome 9 and 22 get fused, resulting in an enzyme that's constitutively active. And the BCR-ABL tyrosine kinase activity is then inhibited by the tyrosine kinase inhibitor Gleevec. So here we see on chromosome 22 and chromosome 9, there's a translocation leading to the fused BCR-ABL gene. And here you can actually see on the chromosomes for chromosome 9, it becomes larger, the mutated. For chromosome 22, the mutated becomes smaller. And that's illustrated here in the cartoon. So the BCR-ABL then is constitutively active, and these patients then respond to Gleevec. There's always some toxicity, especially rash, using these tyrosine kinase inhibitors. And after a year on Gleevec, 53 out of 54 patients responded initially. 51% of these patients were still doing well after a year. After 5 years, 89% of the patients still responded, but some of them had developed resistance to the drug. And so Gleevec, it's a tyrosine kinase inhibitor shown in purple. And when it binds to the enzyme, the ATP can't get in and phosphorylate protein substrates. So we mentioned then with the Gleevec resistance, it's because of a T315I mutation, and they're now developing new drugs for this mutation, such as ponatinib. So what we're seeing here is that cancer, it's not a static disease, it's a moving target. And you can find a drug that works, and then a year later that same drug doesn't work because the cancer's migrated or mutated into something else. So we see here then for CML, it's the BCR-ABL translocation, and imatinib is primarily used for breast cancer with HER2/neu. The antibody Herceptin is used for melanoma. It's got the BRAF mutations. The PLX4032 is used for GIST. It's the c-KIT mutation. Imatinib can work on that. And for the non-small cell lung cancer, we have mutations in the EGF receptor, and gefitinib and erlotinib are biologically active. So we're going to conclude this lecture on practical steps to prevent cancer. The first thing, check your house for radon. When I first moved to Maryland, I lived in a house out in the country, And when I went to sell this house, I was told it had radon in the basement. So to sell the house, what I had to do was install an air pump that pumps the air out of the basement into the outside atmosphere. And so in Maryland, a lot of the houses on hills especially, they have rock. And the rock has the radon gas in it. So the basements can get contaminated. And I was told that if anyone had lived down there, it would be the equivalent of smoking 2 packs of cigarettes a day. So you can get lung cancer from radon. A second thing, check your house for asbestos, such as the walls have a lot of insulation in them, and the old insulation had asbestos in it. So as long as it stays in the wall, it's fine. But if you start going into that wall, the fibers will get into the air, you can breathe them into your lung, and you can get mesothelioma of the lung. Take precautions in your workplace. My old office was in building 31, and on the walls, they had a little sign, "Beware of asbestos," because this building was built 30 years ago, and that's when they used all the insulation with the asbestos. And it still hasn't been replaced in all of NIH. Check your community water system. You want to be drinking good water, not water that has carcinogens in it. Avoid breathing polluted air. Well, when I was a graduate student, I lived in Los Angeles, and every day around 2 o'clock, the ozone would roll in from Western LA and Pasadena. You'd just start choking. Your lungs would start burning, and then 5 o'clock, it would all blow east to Riverside, and then they would get it. So more and more places now are having polluted air. Protect your skin. We mentioned about the melanoma, and it's ever increasing in Arizona as well as Texas and Florida. One of the primary reasons for this is we normally have ozone in the upper atmosphere that protects us from the UV rays of the sun. But the automobile exhaust depletes the ozone in the atmosphere. So we're at greater risk now than we used to be. Don't breathe smoke. Initially, they found lung cancer in chimney sweeps in England, people who would go up the chimneys and clean off the soot. So all that soot has lots of carcinogens in it. Exercise daily. Obesity leads to colon cancer. I myself am a bicyclist, so in a couple of weeks I'm supposed to do a 100-mile bicycle ride. Avoid pesticides. Pesticides can lead to prostate cancer. Eat fruits and vegetables. These have lots of antioxidants and reducing agents in them, so this prevents the oxidation of potential carcinogens. Reduce red meat consumption. That's got quite a bit of fat in it. Eat fish that have omega fatty acids. Minimize fried foods. You don't want to eat too much of that barbecue and eat a lot of that soot. Drink alcohol in moderation. If too much alcohol can lead to liver cancer. Avoid unnecessary x-rays and reduce infections. Infections can lead to inflammation, and it's thought that almost 18% of the cancers result from inflammation. What happens is the antibodies come in and they release all of these cytokines, and a lot of the cytokines can be used by the cancer cells to grow. And finally, we have a few references for you. So that concludes the introduction. I'd be happy to answer any questions. Yes?
When you mentioned that cancers are mutated that often, what drives resistance, do you have an idea of what fraction of cancers are driven by mutations in genes or driven by mechanisms shut down genes that target?
Well, see, with cancers, you have to recognize, first, a tumor, it's a mixture of cells. And lung cancer, in the 80s, we developed all of these cultures from biopsy specimens. Basically, you get the specimen from the patient, You dissolve it in collagenase, you put it in a serum-free medium, so all the normal cells die, and what's left are the cancer cells. But even so, these cancer cells, there's a mixture. And about 1% of the cancer cells are thought to be stem cells. So what happens in lung cancer especially is we have what's called this field effect. So one clone grows out, and then you may stop this clone, but then a different clone grows out. And the first clone may be sensitive to chemotherapy. The second clone isn't. So then you look for your mutation, such as the EGF receptor. And if you see this second clone has the EGF receptor mutations, you can then use the tyrosine kinase inhibitor, erlotinib. But it's a very peculiar thing. They found using these tyrosine kinase inhibitors, the cancer cells were pretty smart, because after a limited period of time, they could figure out ways around it by causing further mutations. Now the scientists are trying to become smarter. They're developing a second generation and third generation of tyrosine kinase inhibitors. But cancer, it's very clever. It's always mutating, and so it's very difficult to fight. And one of the things that we really have nothing for right now is if it undergoes a KRAS mutation. And so that's why they have this new project in Frederick where they're trying to figure out something to treat patients with who have KRAS mutations. So it's a multitude of things. You not only have the mutations, but there can be silencing of tumor suppressor genes. And this sort of can be switched on and off, whereas the mutation, it's an event that can't really be changed once you get mutations. So you have the epigenetic phenomena also contributing. And so it's a very complex problem, and that's why we're so much into the genetic analysis of the tumors here at NCI.
Yeah, I guess my question is about more, I mean, you're going to miss all the chromatin changes if you only look at genetic analysis, right?
Mm-hmm.
So, I mean, do you have an idea of what fraction of resistance is caused, has an unknown cause? I mean, you said there's one example with
Yes, in lung cancer, when they find the resistance to the tyrosine kinase inhibitor, 50% of the time it's because the EGF receptor has become mutated. 20% of the time it's because then the KRAS became mutated. And about 15% of the time it's because of c-KIT or MET mutations. And then there's about 15% where they have absolutely no idea. So those are hard numbers for lung cancer. And the 15%, we just don't know. But the good news is now we have the immune checkpoint inhibitors, and they're starting to work on some of the lung cancer patients. So we have a lot of hope for that now. So 10 years ago, all we had for lung cancer was chemotherapy and a little bit of radiation therapy. Now we've got tyrosine kinase inhibitors, and now we're starting to get immune checkpoint inhibitors. So we're really quite excited about that. And the lung cancer deaths now, they've leveled off. They're no longer going up in the US. Would like to get it to the point of the breast cancer where the deaths start going down. Yes? Right. Well, the EGF receptor is mutated in about 13% of the non-small cell lung cancer patients. The way they initially figured it out that it was mutated is they started using the erlotinib in the US, and it really didn't work very good in US patients. And they tried it in Japan, and it worked quite good. And then they found out that in the Japanese women, about 30% of them had EGF receptor mutations. And most of these were non-smokers. So then they found out in the US, well, this is only going to work if, the patient has EGF receptor mutations. And so in the US, we have about 10%, but in some of the Asian countries, 30% of the cases have mutated EGF receptor. So those drugs are very much in vogue there. But you know, the thing we still worry about is the cancer stem cells, because we don't really know what they're doing. They're in all of our cultures, and in lung cancer, it's about 1% of the cells on the average are stem cells, and we still don't know what they're doing because, you know, it's all the other cells that have the EGF receptors. We're not quite certain of the properties of the stem cells, but the Notch pathway seems to be involved there. Okay, we'll move on then to our next speaker, and we're very pleased to have Jill Smith here from Georgetown University, and she's going to talk about translational research, bench to bedside, clinical trials. No, it's very hard.
But it doesn't matter which one I point to.
It doesn't. It would be easier if you want to use your mouse to highlight your talk. That way the audience here and on far to recognize exactly what to highlight. Your preference.
And I think can't use this.
Yes. Totally different. Give me 1 second.
I like standing out here. You're going to get a kink in the neck. Well, good afternoon. As he's getting that set up, how many of you in the audience are either PhDs or PhD candidates? So, okay. How many of you are MD or MD candidates? Okay, good. Anybody both? We got, good. All right. All right. Good. Understood. Okay. All right. Well, thank you, Dr. Moody, for inviting me. This is my new home at Georgetown. Last year I was here at NIH. So this is my new place, and I haven't quite figured out on this picture. My lab is somewhere next to the medical dental building. It's called Building D because I understand they haven't gotten a donor yet for it. So I do have a few disclosures. I'm a co-inventor on a few patents, and some of them I may mention some of the science from those patents here that have to do with pancreatic cancer. And I'm also the director of a consulting company that helps with some biotech development. As far as what we're going to talk about in the next less than hour, I hope, is how to understand how an idea is actually taken from the research lab to patient care, and learn about the steps of conducting clinical research, and to understand some of the obstacles that you have to overcome for drug development. And some examples I'll give you from my experience with translational projects. We'll talk a little bit about the pitfalls and also the prizes. So first of all, you guys are all here because you have dreams and ambitions, and we all do, and you want to, you know, discover that cure for cancer. Well, I have dreams too. My dream is that I'm going to get the Nobel Prize someday, and I haven't given up on that yet, and I hope all of you have dreams and ambitions also. And there's a few people that I admire. One of them is Nelson Mandela, and he said there's no easy walk to freedom anywhere. Many of us will have to pass through the valley of the shadow of death again and again before we reach the mountaintop of our desires. And then there's Edmund Hillary, who is also another person I admire. He's the first person who summited Mount Everest. But you know, he didn't make it the first time. The first time that he tried to summit Mount Everest, actually the majority of his crew, including his best friend, died. And when he was talking to the Parliament in England, there was a big picture of Mount Everest behind him. And he turned around and he looked at this poster and he said, you know, Mount Everest, you have defeated me. but I will return and I will defeat you because you can't get any bigger, but I can. I'm building up to encourage you guys not to get discouraged when your research doesn't work out the first time. So, and then of course there's Martin Luther King Jr., who did have a big dream, and he dreamed that one day this nation would rise up and live out the true meaning of life's creed and hold all these truths to be self-evident, that all men are created equal. So we all have dreams and desires, and we're all here because we want to see what you're working on in the lab and your discovery to eventually make it to have some meaning in life and to help people. There's a problem, though. I mean, many of us are involved with research, and there's a lot of drugs being tested. There's a lot of ideas that are being tested. But unfortunately, there's this bottleneck. And of all of the ideas and great things that we're working on in the lab, only a certain percentage of them actually get through to clinical trials. And only a certain percentage of those make it and get approved by the FDA for treatment. So what's the bottleneck? This is 495 about now, close to the 5 o'clock rush hour traffic. And is the bottleneck because, you know, all these ideas are crazy, or there's a lot of politics involved with this, and you got to get FDA approval, and you've got to please everybody, and the congressmen have to agree, and whatever. So there are some bottlenecks that are political, too. So several years ago, NIH actually set up the NCATs, and they had some ideas about how they might help improve translational science, and some of them are shown up here on this slide. And they really wanted to focus on these main 6 areas that might help improve taking ideas from the lab to the patient care. Well, where do you start? And this is where you are. Okay, you're the brilliant grad student or postdoc sitting in the lab. You get a wonderful, brilliant idea. And then, you know, of course, you have to think about this idea, and this is the idea downloading. So you've got to come up with an idea, and then once you have the idea, you have to have a hypothesis. So what is this idea that you have, and what's the hypothesis that you plan to test? Well, there's different phases of clinical trials, and most of the trials involve new drugs, and they go through a series of steps. Many of you are working at the stage where you're in the laboratory. You may be working with cells in vitro or cancer cells. You might be working with animals and testing a drug or a new treatment on a tumor or a knockout animal. And then once it's deemed safe in animals, then you go through different phases, phase 1 through 4, and that's testing it in humans before it actually then will be approved by the FDA. So there's also several different types of clinical trials. There's the treatment trials, there's prevention trials, early detection like biomarker trials, screening trials, diagnostic trials, quality of life trials if you're into psychology and clinical benefit. And today, as Dr. Moody talked about, there's a lot of genetic trials that are going on based upon what we know about the new genome. Also, it can be phase 1 through 4. It can be randomized or it can be non-randomized. It can be blinded or open. It can have a placebo control or maybe not. And it can be a pilot trial or maybe not a pilot trial. So just to review for you what some of these different phases are, we'll start with the phase 1 trial. And phase 1 is when you test it in a small number of people, and the whole purpose is to determine whether your treatment is safe. So phase 1 is primarily for safety and toxicity. And then you want to also decide how the treatment should be given. Some of phase 1 is you're trying to decide, is it better to give it subcutaneously? Can you give it orally? What is the bioavailability? And therefore you do a lot of pharmacokinetics and pharmacodistribution studies to try to evaluate how is this drug metabolized in human subjects. And then what will the side effects be? So mainly safety and toxicity. but it doesn't really tell you anything about how well it's going to work. And you actually may be doing phase 1 in normal people, not with cancer drugs, but some of the drugs we do. Then phase 2 is when you get into the efficacy. And you want to know, okay, we know it's safe in humans. Does it really work? And so the number of people that are tested in a phase 2 trial are a little bit greater. Usually phase 2 trials are blinded, randomized with a placebo. You want to know, does it work? And is your treatment better than the placebo? But in the phase 2, you usually don't compare it to other treatments at that point. However, I will say that, you know, in cancer treatments, sometimes we compare to the standard of care. And when we do that, we look at either an equivalency trial rather than or a superiority trial. But a lot of the drugs are approved just because they're shown to be equivalent to another FDA-approved drug, but they may have a better safety profile. So. And then a phase 3 trial is when it takes a lot of people to do a phase 3 trial, and there's an equal chance to be assigned to one of the two groups, and it looks at how the new treatment then compares to the standard treatment, or you can also compare it to a placebo, but it's a lot larger study. Phase 4 is usually after the drug has been released and it's out in the market. It's usually a post-marketing trial, and it involves thousands of patients. For example, they want to know, now that they have this vaccination approved, how is it going to decrease the incidence of, say, hepatitis B throughout the world or whatever. So they do post-marketing trials. So this kind of summarizes the different phases of the trial, how long they take, what the purpose of the trial is, and the number of drugs that actually get to that point. So you can see that once you get up to phase 3, the number of drugs actually starts to drop off. So a lot of drugs, once they get into phase 1, they find out that they're not safe and they can't go any farther. Or you go to phase 2 and there's no difference than a placebo. So they don't move on any further. So those are a lot of the bottleneck and the weeding out of the drugs to find out which ones actually work. Then with the randomized trials, there's an equal chance to be assigned to one group or the other. One gets the most widely accepted treatment and like the standard treatment, and the other gets a placebo. The other one may get a standard of care. And then the groups need to be fairly well balanced. You can't have like all women in one group and men in the other group. So you try to balance the two groups. So with randomization, you get a control group, and then you have a treatment group. And as I mentioned, this group can either be placebo or the standard of care, because we usually don't like to test our new cancer drugs in normal people, and you don't also like to withhold therapy from patients by giving them a placebo if they have known cancer. So often we will when we randomize, they'll get randomized to standard of care versus the new drug. So what's a pilot trial, and when would you want to do a pilot trial? And I'm working on one of them right now, and usually it's a small study, and it's your first venture into an area, and it helps you kind of iron out the difficulties that are involved with doing a bigger trial. And the other important part about doing a pilot trial is it gives you some idea of how patients might respond to the treatment, and then you can have your statistician use that information to calculate the sample size that you need to do the larger phase 2 trial. So often a pilot trial is an open label study where you're just treating everyone and you want to know how many of them respond. And then you use that information for the phase 2 trial. So, and again, these are treatment trials, and it's to determine the most effective treatment for patients who have cancer, to test the safety and the efficacy of new drugs and the interventions for cancer drugs. And I'm not going to spend time on all the treatment trials and whatever, but the early detection trials, there's a lot going on now looking for different biomarkers, and they call them the liquid biopsies. If you can take a blood sample from somebody and try to make a diagnosis of cancer. So the early detection trials are really important right now, and if you can actually intervene at an earlier phase and maybe prevent cancer or prevent the bad outcomes that we have from some of the cancers. So then there are some other studies like quality of life studies that we do, supportive care studies, and then there's the genetic trials. With the genetic trials and a lot of the GWAS studies, it's become important that we realize that there are ethical considerations. And there's a lot of these huge tumor banks where blood samples have been banked away. Now we know that the IRB requires, if you're doing a new study, you have to have special language in there that talks about that you're going to be testing the person's blood or tissue for genetics. So just keep that in mind if you're doing any of these mutation studies, because that's now required. And how are the patient's rights protected? Well, of course, there's ethical and legal codes that govern the medical practice. And there are review boards that Dr. Moody talked about that you can sit in on. There's the institutional review board. And in order to do a clinical trial, you have to be approved and take the city training, which is an online training, to get approved so that you can be involved in human subject research. There are also data safety monitoring boards that actually monitor the conduct of the study while it's going on. So how do you do it? What are the nuts and bolts, and how do you actually take your idea in the lab to a phase 1-2 trial or get a drug approved? So I'm just going to share with you a little bit of what I do and how I've done it with some examples. And the first thing you need to do, and since this is from the NCI, we're talking about cancer, you need to think about what target you're going to look at. And there's plenty of targets up here, and you can choose your favorite target, whether it's in signaling or whether it's in a receptor, or you can choose a different sort of pathway like autophagy or apoptosis, and you're going to work on a pathway. Or you can pick a particular cancer itself. And, you're going to focus on breast cancer or lung cancer or whatever. So, I mean, my research is focused primarily on growth receptors on cancer, and I also focus mostly on pancreatic cancer. So these are just some of the faces of pancreatic cancer, and, we can see how old the audience is. I'm sure that most of you guys know these two guys down here. Does anybody know who they are? And who's this guy? Okay, good. But getting better. Does anybody know who this guy is? Who? That's Dizzy Gillespie. He's a jazz musician. Yeah, Herman Munster. Julia Prowse was the legs person. Jack Benny and Michael Landon. So those are just some of the faces that we're all familiar with of pancreatic cancer. And the reason that I became interested in pancreatic cancer, and you have to identify with whatever you're working on, whatever field, what's the problem? That's where you come up with the idea. There's a problem here, and I'm going to fix it. Well, the problem with pancreatic cancer is that the survival from pancreatic cancer has not improved in over 50 years. In spite of all of our technology, in spite of all the GWAS studies, in spite of all the MRIs and CAT scans and all the technology we have, there has been no improvement. Why? Well, if you look on this picture, are we going the wrong way? There's one car on this highway that's driving in the wrong direction. So the problem is that there's not been any improvement in survival. And over 70 different chemotherapeutic regimens have been thrown at pancreatic cancer, which is thought to be, this silent killer. People, when they're diagnosed, and they're dead within a few months. But the problem with that is that the chemotherapy has been non-selective and non-specific for pancreatic cancer. And in the end, pancreatic cancer is resistant to chemotherapy, and it wins out. So because of this, Congress has actually named pancreatic cancers as one of the recalcitrant malignancies, and they've actually asked the NIH to improve funding for pancreatic cancer because of this. There was a recent article published in Cancer Research 2 months ago that showed that by the year 2020, pancreatic cancer will surpass breast and colon cancer, and after lung cancer will be the #2 killer in the United States if we don't do something. So nothing's changed for 50 years. And if you keep doing the same thing, and for those of you who are into cloning in the lab, you know if you keep doing the same thing, you're going to get the same results. So we need to change our strategy with this disease. So that's where, I came in and said, there's a problem here. Okay, so what are you going to do? When you're doing research, it's really important. You always wonder, why do I have to take all those basic science courses, and why do I have to understand that and whatever? Well, you do have to understand that. The med students, why do you need gross anatomy? But you have to understand physiology and biology in order to understand the normal before you can understand the abnormal. So in the area that I'm working on with growth receptors, there's a couple of gastrointestinal peptides that involve growth of the gastrointestinal tract. One of them is called cholecystokinin, or CCK. And just to review for you, since I'm sure you're really up on the physiology of the GI tract, CCK is released from the eye cells of the duodenum when there's fatty acids and certain amino acids in the duodenum. And it is a classic hormone. So when it's released, it gets into the blood system, and then it circulates in the blood, and it acts on receptors that are in the pancreas. And when it does that, it releases digestive enzymes that helps you digest your food. The other thing that it does is that it reacts to the receptors that are on your sphincter of Oddi and in the gallbladder, so it causes contraction of the gallbladder so that the bile is released, and it helps you emulsify the fat so that you can digest your fat. So this is what CCK normally does. So it releases digestive enzymes. It also regulates insulin release. But the one area that I was interested in, and with my mentor, he was working on growth and regeneration of the pancreas. And they knew that the CCK actually worked on its receptor to help the normal pancreas grow. And if you have a bout of pancreatitis and there's injury, CCK acts to restore the normal cells. The other related gastrointestinal peptide that's in the same family and acts on the same receptors is gastrin. And you all may be familiar with gastrin because it stimulates the release of gastric acid. Well, gastrin was actually present in the fetal pancreas, and it's responsible for growth and development of the normal pancreas, but it's shut off at week 14. However, it gets turned back on again in cancer and actually stimulates the growth of cancer. It also stimulates the growth of the stomach. And for any of you who take chronic proton pump inhibitors, and I don't want to scare anybody in the audience, but omeprazole, all of these other drugs, I don't want to name them all, but any of the drugs that block proton pump in the stomach to block acid release, over time, what happens is that gastrin levels can be elevated because the body is trying to, it sees that there's not enough acid there, so you block the normal feedback loop. And I'm a gastroenterologist, so typically what we see in people who've been on chronic proton pump inhibitors for a long time is they grow these polyps in their stomach. They're benign polyps, and sometimes, you can actually grow other types of things like carcinoids and other lesions. But that's because gastrin and CCK act on the CCK receptors, and they cause growth. So if they're elevated, they stimulate growth. So what does CCK do besides that? The other thing is that CCK can cause pancreatitis. And one of the things that Dr. Moody talked about is inflammation can predispose to cancer. Well, there's a compound that's on the skin of this pretty green frog. It's called cerulean, but it's actually a CCK peptide. It's a decapeptide. And if you take that CCK or cerulean and you inject it into your laboratory animal, you will get pancreatitis. And if you continue to do that over a long period of time, you can get chronic pancreatitis, which can increase the risk for pancreatic cancer. So if you have a little bit of CCK and it helps you digest your food and contract your gallbladder, that's good, just like a little bit of wine might be good for you. But if you have a little bit too much, these are my liver patients, you know. Too much CCK is not good, and it may stimulate chronic pancreatitis and pancreatic cancer. So, but the other thing that I wanted to tell you, not only does CCK can cause pancreatitis by stimulating the receptor, but it has another property. And how does it cause growth? In order to understand how these peptides cause growth, you have to understand the difference between hypertrophy and hyperplasia. So first of all, an example of hypertrophy, and I like Oprah, but hypertrophy is when you increase in size but not in number. And that happens to many of us when we get to middle age. So that is hypertrophy. This is an example of hyperplasia. So she doubled in, or more than doubled in numbers. So you're increasing the DNA content. So one of the things that they knew a long time ago is that CCK and also gastrin, they cause both hypertrophy and hyperplasia. And if you treat animals with CCK, it increases the DNA content and tritiated thymidine uptake. So it stimulates growth by stimulating DNA synthesis. And, you know, they used to think, well, let's just give the animals this CCK and maybe the inflammation that it's causing in the pancreas is increasing the cancer. But they now know that it's not just because of the inflammation that's induced, it's because CCK has this other property that it causes hyperplasia. So if they give CCK to animals that are also receiving a carcinogen, they find that they can significantly increase the number of animals getting pancreatic cancer by doing this. So that's because CCK has properties of both hyperplasia and causing inflammation, both which can predispose to cancer. So back when I was a postdoc, I said, this is a really interesting idea, and we know that pancreatic cancer doesn't have any treatment. And I tested human pancreatic cancer cells, and I treated them with CCK and with gastrin, and lo and behold, the cancer cells responded to these peptides, and we stimulated the growth of the cancers. And we could block the effect by using receptor antagonists. So this showed that these peptides are driving the growth of pancreatic cancer. So just a little bit about the receptors that are involved. There's 3 main receptors for the CCK receptor, and they're easy to remember, A, B, and C. A was originally discovered in the alimentary tract, so it's called A. B was originally discovered in the brain, so they called it B. And C is discovered in cancer, so that's how you can remember them. And the C receptor is one that our lab discovered, and I'll talk a little bit more about the C receptor. But the receptors themselves are significantly overexpressed in pancreatic cancer, just like estrogen drives breast cancer, androgens drives prostate cancer, gastrointestinal hormones drive GI cancers. And compared to the normal pancreas, you can see that the number of the receptors are significantly increased on pancreatic cancer. So what happens in the normal situation, if this is a normal pancreas and it has normal receptors on it, and when CCK is released into the circulation or gastrin, it binds to the receptor, and as we learned, it releases enzymes. But in cancer, the receptors are markedly overexpressed. Some of them have this spliced variant of receptor, which I'll mention in a minute. And when they respond to the ligands, they don't release digestive enzymes, because cancers don't care if you digest your food, but they proliferate. So that's their mechanism. And not only that, but then gastrin, as I mentioned, gets turned back on. It was originally in the cancer cells and it's suppressed, and the messenger RNA gets turned back on in early precancerous lesions, and it regulates its own growth by an autocrine mechanism. So one of the things that we discovered is that not only are the receptors overexpressed, but some pancreatic cancers actually make a mutant receptor called the CCK4 receptor. And it's a spliced variant receptor where the fourth intron is not spliced out. Now, this only happens in humans because humans have an open reading frame through the fourth intron, whereas animals, mice and rabbits in particular, have a stop codon, so they do not transcribe this intron. But what we found was that this occurs in over 35% of the people who have pancreatic cancer. And I won't go into all the details, but we did some DNA sequencing and we actually identified the SNP that causes this spliced variant. And for the people who have this splice variant, when they don't splice out the fourth intron, it causes the addition of 69 amino acids to be added to the third intracellular loop. And for any of you who work on G protein coupled receptors, you know that the third intracellular loop is involved in GTP protein coupling, and that's what causes the growth and proliferation. So when this additional piece is added on here, it makes the receptor constitutively active. So we actually raised an antibody against this specific receptor, and we found that we could get staining in the people who had the A SNP, but in people who had the wild genotype, they did not have any binding to our antibody. So one of the things that I'm working on, which I'm still translating into humans, is we've made this into, it's a monoclonal antibody, and hopefully we'll be able to use this not only for identification, but maybe for treatment of patients. Because we know that if we treat the mice with this monoclonal antibody, the red bars are the mice, and you can see that the monoclonal antibody inhibits growth, whereas those that were treated with controls continued to grow. So, and this just is an example of how it works in that we found that the SNP causes a misbinding to a splicing protein. So if you have the C variant, then it doesn't bind to the splicing, it does bind to the splicing protein, but if you have the A genotype or the SNP, there is no binding to the splicing protein, so you get missplicing of the intron. And that's just another example of it. But is this clinically relevant? Well, we did go back and we did an analysis, which is another type of research you can do. And we went into our tumor data bank and we analyzed patients who had pancreatic cancer. And we said, well, we know that if you have this spliced variant, it seems to stimulate growth. Does that really affect the survival of patients? And indeed, we found that it did. So people who had the spliced variant receptor did not live as long, and it was statistically significant. So this may be one of the reasons why patients with pancreatic cancer have such a poor survival. So now we've identified it's actually a germline mutation for pancreatic cancer. And you have to, of course, correct for the stage of disease, which we did, and showed that it was still statistically significant. So the other receptor that I worked on, since my work has been with G protein coupled receptors, is with an opioid receptor, which is also a G protein coupled receptor. And we identified a protein, OGF, as we called it, that when it binds to its receptor, it inhibits growth of pancreatic cancer. And we did receptor binding studies to show that it was physiologically relevant. And then you can see in these nice nude mice that if we treated the mice with this OGF, you can see that their tumors shrunk, and that was giving 5 milligrams per kilogram 3 times a day. So we were next faced with the idea, okay, I want to do a phase 1 trial. How much do I give to a human being? So, you have to calculate, well, if this is how much I give to my 22-gram mouse, how much do I have to give to my 70-kilogram man? And now I'm going to decrease that by like 1,000-fold and start low and work my way up. So, so we had identified the problem that pancreatic cancer had a dismal prognosis. Our hypothesis was that the OGF could inhibit the growth in the cancer cells and the mice, but could it work in humans? So we went into a phase 1 trial. But before you do the phase 1 trial, you have to apply to the FDA. You have to get an investigational new drug number or an IND number, and you have to fill out what they call a 1571 and a 1572 form. You write a protocol, you get a consent form, you have to get IRB approval, you find a sponsor who's going to fund your study. That's probably the hardest part. And then, you know, you have different responsibilities of the principal investigator, and you have to register your trial on the clinicaltrials.gov website. If you do not register your trial before you enroll your first patient, you are not allowed to publish your research. So remember that. So this is an example of what a 1571 form looks like. And you get assigned an IND number, and you have to include that number with every single time you submit a form to the FDA. This form has to accompany every communication to the FDA concerning your protocol. And then every time you submit it, you have to keep a record of, this is number 1 that I submitted, this is my second communication, and you're given a serial number. And the FDA records all of these. But it's very important to keep good records with your FDA. So then when you do these are the things that you have to put on the 1571 and 1572 form, and just some of the details about that. And then you go into what the aims of the study are. So we wanted to look at the safety and toxicity of OGF. We didn't know what dose to use. So we wanted to calculate what was the right dose. We did pharmacokinetics. We didn't know if we could give it better by IV versus sub-Q, so we tested both. And then once we figured out a dose, we tested it chronically to see how well it was tolerated over time. So when you're doing a phase 1 trial, you have to decide, first of all, who's going to be your population. And a lot of phase 1 trials are done in healthy controls. Well, since we were interested in getting information on cancer patients, we tested this in pancreatic cancer patients. And you just come up with the criteria that you think that will be good, include who you're going to include in your study and who you're going to exclude from your study. And basically, you want to make sure that you're going to hopefully include patients that are going to live long enough so that you'll get some information from the study. What we did in our phase 1 study, which is very typical, is a dose escalation classic 3x3 design, where you enroll 3 patients into the study, you give them the first dose, and we actually started at 25 milligrams per kilogram. And then if the 3 people tolerate that dose, then you move up to the next dose. Well, the very first person that I gave this drug to was a little lady with pancreatic cancer, and she ended up in the hospital that weekend with abdominal pain. And I had to make a decision, wow, did I do this to her, or is it her cancer? Am I going to dose the next couple patients? And I ended up going ahead and dosing the next couple patients. And if I hadn't taken a risk or moved ahead, this study would have never gone further. So she did fine and recovered, and it was probably her cancer that caused her to be that way, because no one else had that problem. But we did escalate, and we found out that 250 was actually our maximum tolerated dose. We did pharmacokinetic studies where we measured and we could prove that it was increasing in the blood, and sometimes you need to do this in your phase 1 trials. And then when we completed the phase 1 trial and we knew what dose seemed to be the maximum tolerated dose, then we went into a phase 2 trial. And you want to know phase 2, as I said before, is efficacy. Does it really work? Well, we ended up doing an open-labeled study, and we used the dose that we had found in the patients in the phase 1 study. Our patients couldn't be resectable. And this is where I went round and round with the FDA because, you know, I wanted to compare it to standard of care, but they said that I couldn't do that because we didn't prove yet that it was efficacious. So the FDA made us treat patients who had already failed therapy, which, if you know anything about pancreatic cancer, you know, they're not doing too well at that stage. But we took what we could get, and we did the study. And actually, we looked at survival in those patients. We looked at other outcomes like time to progression, quality of life, clinical benefit, which is something else we look at often in cancer studies. And we looked at CAT scans. There's the RECIST criteria just to show if you're having shrinkage of the tumors by measuring them periodically on CAT scans. And actually what we did find with our primary endpoint of survival, that compared to patients who chose not to go in this study, that we had significant improvement in survival of our patients who had this treatment. So after we did the phase 2 trial, we actually then went back to the lab, and we thought we would test it in combination with another standard chemotherapy. At that time, gemcitabine was standard treatment for pancreatic cancer. And what we found is that when we gave the gemcitabine alone or OGF alone, the tumors didn't shrink as much as when we gave them in combination. And you can see with the mice over here, if any of you work with nude mice, the mouse that got the chemotherapy actually looked, he lost weight, and his skin was kind of scaly. But the animal who got the combination, it had a protective effect, and he did okay, and the tumor was smaller. So to make a long story short, I went back and applied for another grant and got more approval, and then we tested this combination in human subjects. And we actually found, compared to our own monotherapy with OGF, we could improve the survival of patients who got the combination. So this drug actually, this is when I was at Penn State. Penn State has the intellectual property for this. But this is an important point, because before you go out and disclose any research that you do, including an abstract, you want to make sure that you have filed an invention disclosure or a provisional patent to protect it. If you're working, and the discovery actually belongs to your employer, not to you, unless you own your own company. So anyways, but that's important. With the OGF, we did intellectual property. Penn State got the patent, and they actually have licensed the patent, and it's going on now for development through the FDA. So it is moving forward, and I'm an inventor on the patent, so, you know, eventually I may get royalties, but, you know, that's what happens when you work for a company. But you could also, you know, you have to find a company, you have to license your patent if you really want to get it out there, because most of us are, you get it to a stage, and then you have to find a company that can actually take it the next level. So what are some of the obstacles with doing translational research today? Well, of course, the biggest obstacle is always money. How I did a phase 1 trial with an RO3, which was $50,000, is just unbelievable. But, you know, I had a company give me a little grant that helped me buy the drug, and the other thing we used is the CTSAs that are at the universities, because that helps keep the cost down. So it is possible, but, you know, it is expensive, and funds are hard to come by. The other thing, I mean, I'm a clinician, I'm an MD, and clinicians often don't get protected research time, which it would be nice if they had more protected time. And the indirect costs that we get on our grants don't cover our malpractice. So MDs have to see so many patients to pay their malpractice, because you're required to have malpractice if you're doing clinical research. It's required by the FDA and by the IRB. So you have to somehow pay for that. So that's another problem. There's also this chiasm between industry and NIH and academia. And I think in order for us to move forward with a lot of these drug discoveries, that chiasm has to come to a close, It's all about team science today, and we all got to work together. And you know, I think that, you know, industry has a lot to offer. Academia, NIH has a lot to offer, but they all need to get together and work together if we're going to come up. And like I said, there's no more one-man bands. This is all about team science. So as we look back on the problem, you know, survival of pancreatic cancer, why hasn't it improved? You know, are we all going the wrong way? Well, maybe you got to take a different strategy. Maybe actually this guy is going the right way and everybody else is going the wrong way on the highway. So in order to change things with research, I suggest that we have to transform our minds. We have to think outside of the box. We got to do things differently or else things aren't going to change. And don't be afraid to take some risks. If I hadn't moved on and pushed forward, we never would have gotten that drug moving through an approval through the FDA. So you have to be able to take some risks. And the bottom line is does research have any real clinical relevance? Does it help people? These are a couple of my long-term survivors from the pancreatic cancer studies, and I have their permission to show their pictures, Bobby and Vicky. And yes, it means everything because of people like this. So that's the reason why we're doing the research, is to improve survival and help save lives. So the important thing is don't give up. And I'm here to tell you that. You know, I haven't won the Nobel Prize yet, but I haven't given up yet. So if it's not me, I hope it's one of my students or somebody else. But as far as your dreams, there's some sayings. One is that strivers achieve what dreamers believe. You got to work for it. There's also a saying that says, I stand for freedom of expression, doing what you believe in and going after your dreams. And a dream doesn't become reality through magic. It takes sweat, determination, and hard work. And this is what I said, you know, if you don't believe in yourself or your dreams, no one else is going to believe in them either. So you have to believe in yourself. You have to have the faith in your work. And most of all, don't give up. Be encouraged. So I want to thank Dr. Moody and Dr. Zia and some of my colleagues for helping with all this research. And thank you for your attention. So. Questions?
Yeah, similar to the OGF
So it does. So the OGF receptor is a nuclear receptor, and so there's the three classic opioid receptors, the mu, kappa, delta, that are on the plasma membrane. The opioid growth factor receptor is on the nucleus, and it actually, when the OGF binds to it, it shuts down DNA synthesis. 5 amino acids. Small, so it can penetrate the cells. Entertaining it, yeah, that would be good, yeah. So. Mm-hmm. I want to do that, and that's coming in the future. The paper is still in press, hopefully to come out in a couple, it takes so long, I want to say a couple weeks, that we discover this splice variant. But now that we know about it, I mean, it would be ideal if you could screen for patients and you know who might be at risk for developing pancreatic cancer. Because right now we're just looking at people who have familial pancreatic cancer, which only makes up 10% of all the pancreatic cancers. So one, if you know that you have the mutation, kind of like the BRCA2, you might be able to improve surveillance and follow people more closely. Also, if we know that they have it and we have this monoclonal antibody and we can target the receptor, as Dr. Moody was talking about, now that we know different mutations and things, we can start doing target-specific therapy. And I think that's where the future is going with all this treatment. Yes?
How would you characterize
the maximum tolerated dose is determined based upon the side effects and the toxicity that you get with your patients. So you escalate it to a certain dose, and when you get to the dose, I mean, certainly if you kill somebody, you don't want to use that dose. Then you have to back down to the next dose prior to that. There's different, and you design the protocol, but you look at the different things that you would expect for toxicity, and if you get those toxic side effects, then you know that you have to back down. Okay, so that's what you do. You go up to a level where you find that it's well tolerated, the majority of the people will tolerate this, you don't have the serious adverse events, and then that's it. If you do get a serious one, you have to back down to the prior dose. Yes? Of the OGF? The OGF is actually a peptide, so we gave it by infusion. And we found that we could measure it in the blood. It had its peak within 30 minutes in the blood by far. Is this what you're asking? No, maybe not. Okay. Oh, okay. The median overall survival of people who are treated with this. So most of our patients were people who had prior therapy. And so in monotherapy in those patients, we had survival up to 9 months. And in our patients with combination, it's more than 11 months, which is the current thing with FOLFIRINOX. Are they what? Yeah, so in the only study that we did when they were treatment naive was the combination, where we gave it along with gemcitabine. And that's the one that we had several patients living several years, okay, with the combination therapy. But we also were using patients who hadn't developed drug resistance and they hadn't already failed other therapies. So in our monotherapy study, you know, the survival was 9.1 months, but some of those patients had already failed prior therapy. So, you know, ideally, if you could use this drug from the onset and maybe give it with current therapy, whether you're using Abraxane or FOLFIRINOX, it may improve survival and decrease toxicity, which is what we were seeing. And they won't, I mean, they act by different mechanisms, so that's why they potentiate each other. Yes, in a way. When we went into our study, I wanted to do it like as a phase 3 study. And the FDA actually made us go back and do a phase 1 study because they said that we hadn't yet looked at the safety and toxicity of the two drugs in combination. So, you know, so there are certain rules you have to go back because, you know, nobody knew that if you gave gemcitabine with this compound, was it going to make it better or worse, you know. So, so they do have certain requirements, and unfortunately it sometimes slows things down, but they do. Yes? Right. I mean, that's a great idea. You want to come work in my lab and we'll figure it out. Yeah, I mean, as Dr. Moody said, the stem cells become resistant, and that's a problem with recurrence. With pancreatic cancer right now, most people survive only 3 to 6 months. I mean, we're happy, you know, the latest and greatest treatment with Abraxane, you know, with gemcitabine is up to 8.1 months or something. And with FOLFIRINOX, 11 months. I mean, we're not even to a year yet. So, I mean, we're not talking about recurrences. We would just like to get someone to regress, you know. So, but I think that's a very important point about the stem cells, because we're going to have to make sure that we do attack the stem cells so that when we get to the point where we have effective therapies, they don't keep coming back or they don't get resistant. So, but yeah. Okay, well, thanks for your attention.